So yeah, we'll get rolling here. Welcome everyone. My name is Matt Miller. I'm the general manager of the Medway Community Forest Co-op, and we're hosting the second in our series of webinars centered on integrated pest management for Hemlock Woolly Adelgid. Before we get started, I just want to start with the land acknowledgement and acknowledge that we are meeting here today in Guestwick, one of the 7 districts of Mi'kma'ki, the ancestral and unceded territory of the Mi'kmaq people. This territory is covered by the treaties of peace and friendship, which the Mi'kmaq and Wolastoqey people first signed with the British Crown in 1726. The treaties did not deal with surrender of the lands and resources, but in fact recognized Mi'kmaq and land title and established the rules for what was to be an ongoing relationship between nations. We'll note that the term public lands and crown lands are complicated topics in and of themselves. Canada as a nation was built on lands that were already occupied by Indigenous peoples who were living on their own lands under their own laws and in their own societies at the time of discovery and colonization by Europeans. We acknowledge the Mi'kmaq as the past, present, and future caretakers of this land and thank them for sharing their homeland with us. We are all treaty people. So, we're really lucky tonight to be joined by Kirk Hillier and Jeff Fidgen. Kirk Hillier is a professor of biology at Acadia University and director of the Insecta and the Acadia Agritech Laboratory. His research program focuses on the development of sustainable technologies to manage insects that affect agriculture, forests, and human health. More recently, his work has focused on biological control and risk assessment, strategies for managing invasive forest pests, including Hemlock Woolly Adelgid, with ongoing research on predator releases, non-target impacts, and ecosystem level outcomes in Eastern Canada. We've also got Jeff Fidgen, is a biologist with the Canadian Forest Service with over 33 years of experience in forest entomology, 25 of which have been spent researching Hemlock Woolly Adelgid. He is part of a large research team at CFS studying HWA. Recently, he has been collecting, releasing, and evaluating introduced predators of the Adelgid that will hopefully give hemlock a fighting chance against HWA in Nova Scotia. So yeah, very lucky to have these gentlemen with us tonight. They're going to give us an overview of biocontrol programs here in Nova Scotia and how this piece fits into the whole integrated management approach for HWA. If you have any questions, feel free to just put them in the chat and we'll have some time at the end. And certainly when we do get to the end, if you want to ask questions as well, just put up your, raise your hand and we will, we'll get to you in time. So I won't take up any more time. Kirk, we'll have you lead off, and yeah, thanks again everyone for joining us.
All right, thanks so much, and thanks for having us, and thanks for everybody coming out tonight for this. It's always fun to have an opportunity to talk a bit about our work. And I'm looking at the title of my slides. I'm realizing I forgot to put HWA in there, so it should be actually understanding HWA biological control and research in Nova Scotia. But suffice to say, hopefully Jeff and I will iron that out before the end of the evening. So we already had a great land acknowledgement. The one thing I will add is that I did also want to acknowledge that some of this work is engaged on the Pacific coast of BC. So we have additional territories of First Nations, including the Salish, the Nuu-chah-nulth, and the Kwakwakwa nations whose stewardship continues to shape these environments. So again, just in addition to work being conducted in Mi'kma'ki, I did want to acknowledge work happening on the West Coast as well. And you'll hear a bit more about that in Jeff's talk too. We're here to talk about HWA. If any of you were at last week's talk, you're probably familiar with this, and unfortunately, more and more of us are very familiar with this in Nova Scotia. Invasive pest recently found in Nova Scotia and is spreading incredibly rapidly through the province. This is a significant threat to the hemlocks and to the forests in general in Nova Scotia. The East Coast invasive strain of this has basically been on in the eastern seaboard since 1911, and it can kill a tree in roughly 4, but maybe upwards of 5 to 10 years as well. It has kind of an unusual life cycle. If you're familiar with like aphids, these have a sort of a similar life cycle where they're basically nymphs for part of the year, and then they become adults and they move on and create a second cycle of crawlers. They're all asexual females in the east. Originating from Japan, where they actually had a spruce host where males were actually produced. We don't have that here. An unusual thing is that a lot of the activities happening throughout the winter months. So you can see adelgids out on the trees now. These will become adults in March and coming up pretty soon. And then we'll actually see eggs moving forward into the second generation. The two generations being the cystins, present in the winter months, and then the progredens that's sort of in the spring, moving in the summer. Adelgids spread pretty rapidly. Again, things like birds are moving them around as well. Movement of timber and foliage and things can move them around as well. I won't get into the life cycle in a lot of detail here. Suffice to say, it has two native quote-unquote ranges, one being in Asia, where there are multiple populations of HWA that have been isolated by folks from the USDA or the US Forest Service. But we also have sort of a naturalized population in Western North America that's been there for a very long time. And then we have the invaded range on the East Coast. And this is important because these native ranges in Western North America and, of course, in Asia, these have native complexes of predators and parasitoids and things like this that actually help manage these pest species that we actually do not have on the East Coast or not naturally occurring on the East Coast. I don't think I need to tell people too many things about hemlock. It's an important species, but it supports wildlife, water quality. There are wood products. It's maybe not as important commercially as some other forest products, but it is, of course, important culturally and economically, and it provides shelter for animals, prevents soil erosion, maintains water quality. So there's lots of reasons why we want to maintain hemlocks on the land, especially these giant hemlocks that we have in Nova Scotia. It occupies a decent amount of the forest. We're looking at about 8% of the forest, or 216,000 hectares of hemlock. So that's a lot of hemlock to protect. As Matt mentioned, there's a different coverage of this through parks, crown land and whatnot. But of course, a lot of this actually is present on private land as well. And you'll see a little later how far HWA has proceeded throughout this range as well. Again, most of you are probably familiar with this, but HWA can be found in the environment by looking for sort of like this frosting that you actually see on branches. These are actually the ovisacs of individual adelgids that are present at the base of each needle on these hemlocks. And of course, they're feeding by sucking the fluids out of these plants. So they kind of simulate or exacerbate drought, which you can imagine is at least last year was already a big issue for some of these trees. And of course, the impact of this is that we see the trees ultimately dying, loss of canopy, and of course, dead trees overall. Recent distribution of HWA. So we can see in green here, this is sort of the range of hemlock that we see in Eastern North America, Eastern hemlock specifically. And then we can see in brown the areas that are actually infested with HWA, of course, extending up into all the way up into Halifax County now in Nova Scotia as well. So again, you can see that it has spread throughout this range, throughout the southeast United States, and there's really nothing to believe that it can't occupy all of the range of hemlock. So there's really no, boundary layer with climate change that will basically protect hemlock at a higher latitude than we could potentially see HWA occupying. In terms of the adelgid, again, this again just shows the distribution and sort of how quickly it has spread over the last 70 years or so, with, again, the more recent invasion happening in Nova Scotia here, but also in Michigan and also into Ontario. If we will look a little more closely at CFIA's records of hits here, where we're seeing Hemlock Woolly Adelgid being picked up, again, throughout all of Southwest Nova, we see infestations, and then it's been picked up more recently in Hants and Halifax County. Again, that may have changed already since the last time this map has been updated. There are different ways of managing HWA. As you noted, it's been present throughout the United States. So lots of people have been looking at lots of ways of managing this. Things like providing, protecting genetic resources by creating seed banks. Silviculture, so cutting out trees and putting in something more resilient. Trying to develop trees that are resistant to HWA. But really, we're going to focus mostly on biological control. And again, if you attended last week, you would have heard a bit more about the chemical controls that are possible. I'll touch a little bit on some research happening at Acadia related to this, but the majority of what we'll talk about today are biological control agents. So at Acadia, at least, we have projects that are investigating this from a few different angles, either the application of insecticide, which again, you apply to trees either through a basal bark spray or tree injection to provide relatively good protection from HWA for a series of years. But of course, this is an expensive prospect and there's a lot of manpower required to get this on the ground. Second technique is to develop biological controls so we can augment and bring in biological control agents. And as part of that, Acadia has been partnering with organizations like CFS, Parks Canada, and the province as well to develop a biological control facility that's going to help facilitate these biological control agents. I'll touch a little bit on the research on the insecticides because again, just because it's relevant to some of the work we're doing at Acadia. The benefits of the chemical control is that it's quick to deploy. There are concerns about environmental contamination and biodiversity impacts, and of course, it can be expensive. So it's hard to envision treating 216,000 hectares of hemlock. So this is used on high-value stands. In recent years, we have been doing work with Parks Canada, basically, and the Forest Service looking at the impacts of treatments on non-target groups, things like pollinators in the forest. This was done by Luca Voscourt, as well as looking at things like arboreal beetle species or pitfall trapping to look at terrestrial insects to see how these might be impacted by these treatments. And I won't get into a ton of detail on that, but suffice that work is still being conducted to some degree. And we, again, have current research largely with Chris Edge with the Forest Service, but then with a number of partners here as well, looking at non-target impacts as well. And this includes looking at the risk of these treatments. Well, sort of a converse thing. Basically, what happens to the forest, whether you treat or whether you don't treat, because if you don't treat, you're also going to see consequential forest loss. So then we're looking at things like the impact on the canopy, the soil and landscape, and how this gets into things like food chains. 4 different projects for master students have been developed here, one looking at bird biodiversity, one at the forest community of insects broadly, one looking at the pollinator risk of ground-dwelling bees, and then through Mount Allison, there's a project looking at salamander abundance. This work's being done at a number of sites throughout Southwest Nova, either in the Medway area, but then also through watersheds moving down through Southwest or the South Coast, all the way down to McKidgee and Westerly Lake. Within these sites, we have a combination of either sites that were treated with imidacloprid or dinotefuran, imidacloprid on its own or left untreated. And the idea is basically to look at how things like the biodiversity of birds, insects, bees might be impacted in these areas. I won't go into a ton of detail here, but Catherine Potvin, a master student, is actually looking at how the bird community structure may be impacted by either HWA damage or chemical controls using these acoustic recorders and point count surveys of birds. And also whether the birds themselves are actually being subjected to chronic toxic levels of neonicotinoids. So they have been actually out collecting these birds through mist netting and collecting blood as well as diet items to see whether we're seeing high concentrations of these insecticides in the birds. Second project is looking at invertebrate non-target effects. So again, this is Karis McMurray's project. Working in concert at these sites, Karis is basically looking at how insect biodiversity is potentially shifting in, again, indicator species within these environments using an array of different traps at each site. Basically looking at functional yield, so whether insects that are predators versus insects that feed specifically on hemlocks versus insects that may be living at the base of trees might be functional as indicator species of change in the forest, either again through canopy loss or through the chemical treatments. We have another student, Jane Verdula, is looking at sublethal effects on bumblebees, because again, if we think about things like basal bark sprays, these can actually contaminate the soil to some degree. Bumblebees are actually on the ground nesting, but we also have evidence that some of these insecticides can wind up in flowers in the forest too. So the idea behind this is basically looking at whether contamination in the soils impacts bees in a sublethal fashion as well. And that brings me into biocontrol in more detail. So again, we have a number of student projects engaged with this, but outside of the chemical controls, biocontrol is really standing out as the main long-term solution where we're going to see sustainable management of HWA below levels that will actually permit hemlock to remain on the landscape. The issue with biocontrol is that it requires a lot of significant upfront effort. We have to maintain risk assessments and infrastructure to rear the things, but also risk assessments to make sure that they're not going to be dangerous to ecosystems and habitats and communities that are actually present. However, this is likely the only long-term solution. I mean, there's been 50, 60 years of work in the United States looking at different ways of managing HWA, and really things like these silverflies and the Laricobius are really standing out as the primary solutions for this. And again, the benefit is that these tend to be self-sustaining once established. As mentioned, we have populations of these in Western North America, but also in Asia. But to harvest and harness these things, we're basically looking at Western North America. And again, this is important because HWA is spreading. We do not have the native enemies present here. And then again, this was a study that was done through CFS looking at whether there were things already in the environment that could manage them. And of course, this has significant ecological, socioeconomic, and climate change impacts. And again, chemical treatment will only go so far. I'm not saying that we are planning to do this in the absence of chemical treatment. And in fact, chemical treatments need to work hand in hand with biological control. From here, I'm going to basically hand it off to Jeff so that he can comment a bit more on his side of things. So I'm going to stop sharing for a minute. And then I need to change the slideshow and let Jeff take control. So I'll set this up here. And then, Jeff, can you take control here?
Not sure.
Is it responding for you?
Not yet.
Okay. Is there anything on screen for you?
Is there what?
Is the show on screen for you?
No.
Okay. So let's try this again. Share window. As I'm sharing a window. Lovely. Try this one more time. Are you able to just play it from your computer? That might be
You
easier.
should be able to.
Yeah.
That not going to work? I can find it first. Okay. I'm going to share my screen.
Wonderful.
How's that? Can you see my notes?
All good.
Okay, perfect. Well, thanks for having me, everyone. It's been a little while since I've been playing with Hemlock Woolly Adelgid and this little bug here in the corner, Laricobius nigrinus. So what I'd like to do this evening is to give you an update on some of the work that's actually happening on the ground in Nova Scotia with these predators. And so, as Kirk mentioned, most of the predators that we're looking at collecting come from Western North America. They're present in British Columbia, the same species that the Americans are taking from Western US. And so When we looked at the research on these, there's actually 3 predators that we're looking at. I'm only going to deal with one of them, but this one has had the most research done on it. This Laricobius, it's a little beetle. It's very loyal to HWA in the coast of British Columbia. It only eats HWA there. You find it in other adelgids, but it's really not a major predator until you come to Hemlock Woolly Adelgid. And so beginning in 23, we started releasing this insect. And mainly the goal of the releases was to assess whether or not these predators could be used in Nova Scotia with the challenging climates that might come up. You know, we might still get lots of return to good old-fashioned winters. Both the adelgid and the beetle would be very sensitive to very low temperatures that we used to get 20, 30 years ago. But I think with climate change, those are probably in the past for the foreseeable future. And so with the goal of assessing the feasibility of the biocontrol using Laricobius in Nova Scotia, we've developed kind of a workflow here. And I will go through that in the talk. And so we start kind of on it's on my left, I guess, with spatial tools and sourcing. So that would be basically making trips out to British Columbia to collect these predators. And then there's a division here, and Kirk is, I think a little later, is going to talk about the division that goes from sourcing predators to rearing predators at his facility. But we thought we could short circuit this a little bit in the interest of getting predators as quickly as possible in Nova Scotia. So with Laricobius, we can collect those beetles in the fall as adults. And so we can, instead of having to rear them to the adults, we can just take them straight into releases. And then from the releases, once a site is selected for releases and the beetles go out there, it triggers a whole suite of assessments. And as Kirk mentioned, these assessments mainly are to evaluate whether or not the predator can survive in Nova Scotia, but also can it thrive? So can it build up? Can it go on from those points that we introduce it to other infestations in other parts of the forest? In addition, as the biocontrol agents build with time, we can start to look at their impact on the adelgid population, as well as giving the beetle enough time to establish itself in the province. Then we can start to see what kinds of non-target impacts we might be having out there. And the research is showing that, again, that this beetle is actually fairly safe. I say fairly because you can see under non-target effects, there is some hybridization that has been seen. That's so far pretty much the only non-target impact. And I'll cover, maybe I or Kirk will cover a little bit of that later. And so these blue arrows are just basically what I'll cover in this portion of the talk, but really, as you cycle each predator through this workflow, the same things happen. So a bit of an outline. So we're going to talk a little bit about the biocontrol strategy. Now keep in mind that this is very much a research program. So our strategy might be different than the strategy when Kirk is up and running that he'll use on the landscape. I'll go over some of the work that we do out west, as well as some of the releases sites that we have here in Nova Scotia. As well as I'll give you an update on the beetles that we've released over the last 3 years, and then kind of start to dip into a little bit on the post-release monitoring. So this is something that happens after the beetle becomes established. And then I'll spend a bit of time talking about leukotraxis, but just a very bit, because I think Kirk's probably going to cover more of that. And so the biocontrol strategy for HWA overall, not just with this particular insect, but overall, is to target each generation of the adelgid with predators. I'm just going to pause here because I have a window that's covering my slides. Here we go. And so one way we can do that is by introducing predators into the first generation and then introducing later on predators into the second generation. And the reason why we need to have predators on the second generation of the Adelgid is because when you have, now this is without predators in the system. So in Nova Scotia, winter time is a major mortality factor for the Adelgid. So it drops the populations. But what research has shown is that when those populations go down, it actually creates more space and more food for the second generation. So the second generation will produce more offspring, and those have a tendency to rebound the population back to a level before the cold happened. So it's basically very low winter temperatures that do this. And they call it the progrediens bounce. We've called it progrediens rebound, whatever you want to call it, but it's just a way that the Adelgid has kind of a built-in mechanism to be able to survive things like this. I have let's see if I can do it this way. No. Okay. I have a little image here. So the beetle comes in, and he's right about here, and drives this population down any further. But again, without predators over here, this just continues to happen. And so this is where we want to bring the Leucotraxis flies, our specialists on this generation. So if we can get both of them established, then we can really take an honest look at whether or not biocontrol is going to work in Nova Scotia and basically in Eastern Canada. Nova Scotia is the model laboratory, so to speak, for this. And so if you, most of you have traveled around Nova Scotia, so you're not unaware that most of the river systems are lined with hemlock. And we figured that introducing the predators in these riparian ecosystems would help the beetles spread across the landscape. So they'll spread throughout the catchment. They'll go wherever they want to go within Nova Scotia. As I mentioned earlier, there are 4 predators. 3 come from British Columbia, and then 1 from Japan. This beetle, Laricobius osakensis, unrelated to nigrinus, but it's already established in the US. So when we talk about bringing in osakensis from Japan, what we really mean is getting a collection of osakensis from the US that's naturalized, rearing those in containment, and then releasing their offspring in Nova Scotia. So they're very much a homegrown osakensis rather than going to Japan directly and collecting them. But again, I'm going to focus on nigrinus here. Oh, here's the beetles and the flies. It wouldn't work earlier. So a little bit on Laricobius. It is native to the Pacific Northwest. We are collecting just the coast specimens, so the ones that occur basically in the lower mainland of BC. It only reproduces on HWA, and the adults and larvae both eat HWA. It's a winter active insect. It wakes up in late summer, early fall, begins eating HWA. And then in the spring, when HWA begins laying eggs, the beetle starts laying its eggs. Again, there's a low risk of non-target impact with this. In over 30 years of research, that's the only non-target impact they've been able to find with this insect. And so the window here, because the beetle needs to be collected at an adult stage, we're fortunate that in British Columbia and Nova Scotia, the beetles wake up at the same time. So in BC, the beetles are coming out of the soil in October. We go out and we collect them, and we're releasing them in Nova Scotia when the Adelgid is waking up. So it's the perfect timing. And so what do we mean when we say collecting Laricobius? Well, basically, what I'm going to talk about a little bit with this is it's a wide area out in British Columbia. You know, it's probably one of our largest provinces. We need to have ways, part of the research anyway, is to be able to identify areas where the outbreaks of the Hemlock Woolly Adelgid occur more often. And so that would optimize our trips out so that we could focus in on those areas. And where the Adelgid populations are high, we know that the predator populations will be high. So this is where I'll be talking a little bit about the spatial end and the sourcing of the beetle. So over the last several years, I guess since 2019, we've been running around coastal British Columbia looking for hemlock sites. We've been really focusing on the forest that's near the ocean because that's the warmest. It's also a drier location where hemlock really isn't best suited. So the trees tend to be a bit more stressed, and we think that that maybe feeds into why adelgid populations pop up there more frequently. And of course, that's where the beetles are going to be found. Wherever you have a lot of adelgid prey, there's going to be beetle populations building on those. And so this is just another illustration to show what I'm talking about with the coastal Douglas fir forest here in dark green. That is the area where we're collecting most of the beetles. So you can see there's not a lot of it out there, but with climate change, those zones are expanding. We actually are seeing an expansion in the infestations that we find out there. So here's an example of where we've actually collected the beetle. Most of them are down here in Victoria and Souk, which is just to the west of Victoria. And that area has seen significant warming over the last 30, 40 years due to climate change. And so that it stands to reason that if those areas are warmer and drier, that the hemlock are going to be under more stress. They're going to be more prone to outbreaks of Hemlock Woolly Adelgid, and those predators will be there to take advantage of that. And so over the last 3 years, we've collected just over 22,000 beetles from this area. Again, I bring this up just so you can kind of visualize where those sites are in relation to the drier, what they call the coastal Douglas fir forest. So as you can imagine, the adult collections are actually pretty interesting, whereas Kirk will be collecting foliage off hemlock out west and rearing it in his containment facility. We run out to British Columbia and we beet sample the hemlock trees. So the beetles are on the tops of the branches eating adelgid, and then we come along and stick a cloth beet sheet underneath the branch. And we have a white wand, as you see Glenn holding here. And what he's doing, he's going to comb that wand down over the foliage. And when he does that, it dislodges the beetle onto the cloth sheet. Now, a cloth sheet's really important because the beetles grab onto it and hold it. And so you can see this is Jeff Fidgen here running up a ladder. He's the lightest guy of the three of us that were doing it, so we always send him up there. But so the beetles end up on the sheet, and then we're looking for the beetles, and we use these little aspirators to suck them up into a glass vial or a plastic vial. When that vial is getting full, you can see these are the white stuff here is wood shavings. The beetles like to sit in that and hold on to it. But when this jar gets a little full, then we'll transfer it to a bigger cup. And then those cups are brought into in this case, our hotel room. I'm sure the people that come and clean our rooms aren't very pleased with this at times, but we go through the samples and we remove any non-target insects that maybe accidentally were sucked up by the aspirators. And we also look at each Laricobius beetle very closely because the nigrinus out there is jet black. Sick nigrinus can look a little brown, and then there's another species that feeds on larch adelgids that is very light brown. So we don't want to bring those over. We only want to bring nigrinus over. Those cups would get sealed and put in a shipping cooler with a freezer pack, and then shipped directly to a crew in Nova Scotia who grabs those, takes them out to the field to release them. Field releases, now this is in Nova Scotia. So now you can see we're moving through to the release part of this whole strategy. And basically, there's a protocol that was developed at Virginia Tech. I was actually a part of developing this when I was there, which identifies sites based on certain characteristics that would be suitable to the beetle. So as you can imagine, we need to have a lot of lower crown foliage that we can reach by hand to release these beetles on. And that's what we're looking for when we're out there. So not every site that has hemlock is suitable. Typically, we try to find edges where the hemlock branches are more abundant down low. And the reason for that is we need to be able to come back to those sites and sample to see how the Laricobius population is doing. And so if we don't have that foliage, we can't do any follow-up assessments very easily. Now, because Nova Scotia tends to be a little bit more risky in terms of the climate, we have a tendency to select younger trees. That's for various reasons, but we find that these younger stands tend to be a bit thicker in the canopy. We do give preference to sites with a southwest aspect that allows the sun to get in there in the winter and maybe provide a warmer microclimate up in the canopy for these beetles. And then, of course, we're targeting stands with dominant to hemlock when we're doing this, although a nice mix of fir or white pine also works very well. Anyway, I know it's interesting when you read this, you say, why are they putting beetles in very low density HWA populations when we have so many stands out there that are getting hammered by the adelgid? Well, the reason for that is very, very simple. If we were to put beetles in stands that had very high populations of adelgid, once the trees start declining, the adelgid population crashes for a brief time, and that is not good for the beetles. So what we try to do is put beetles in low density stands so that their populations build alongside the adelgid populations. And so that gives those populations enough time to build and spread before the stand would start suffering impacts. And yes, they will suffer impacts. We have way too many adelgids and not enough predators. So for the time being, we will still see impacts despite all the good work that everyone's doing. And again, for this, we focused on crown lands over the period of 23 to 25. One of the requirements, obviously, is no disturbance for 5 years. These beetle populations build very slowly. They have a period of time where they're in the soil as pupae and as adults waiting to come out in the fall. And as you can imagine, in the soil, there's all sorts of insects and other critters that might find them and eat them. So they have a very low survival rate in the soil. And so the populations build very slowly. So we'll move on to some releases. So most of the releases or actually all the releases in 2023 were done on the South Shore. We chose the South Shore because the climate down there is just a bit warmer, and we figured that the winter severity would not be nearly as high. But to hedge our bets here, we picked sites that tended to have good access to water, but also that thick canopy. And also we edged up the population of beetles. So there was some work done earlier that showed that if you release 800 or more beetles at a site with a dodgy climate, so you could get a very cold winter, that 800 or more beetles actually has a higher probability of establishing as compared to releasing fewer beetles. So on the LaHaye site, which is actually in the town of Bridgewater, that was a very warm site. It's always been warm. I checked the temperature data for a lot of these places before we do anything. And so we decided, you know what, let's just try a lower release and see how it works out. And so in 2024, and so go back up here. So the first year, very humble beginnings, about 3,800 beetles were released. So in the next year, we were starting to get pretty good with finding the beetles and collecting the beetles. And so we expanded the number of release sites. So you can see that now, yes, we had quite a few sites on the South Shore, but we're starting to push inland because we really want to give these beetles a good test with potentially warmer or cold, potentially colder sites. Keji has been a great partner in this work. So we have 2 sites established there. The other thing, Westdale up north is even colder, and I won't give it away, but what I will say here is that the first 2 sites on Mersey, where you have 450 beetles each, those were actually releases we did in the spring. So we were out in BC in the spring, we collected in March and released those. One release was done in March, one was done in very early April. And that was designed basically to see if the beetles would do as well as the fall releases. And you can see we're starting to tweak numbers a little bit to try to see if if we do get a bad winter, is that release level high enough that we will still see beetles establishing in the by fall? So you might see here a monster number, a 3,158. That is actually what we call a research sectary. So we put additional beetles there so that anyone that wanted to do research on the beetles could come and make collections and use those. We hope in the future, though, that we might be able to return to this site and collect beetles as population builds up and move those beetles to other parts of Nova Scotia. In addition, we were able to give about 5,000 Acadia for graduate student projects. Kirk will probably talk about that in a bit. So in 2024, about 7,600 beetles were basically present in Nova Scotia. And if you thought that was good, well, in 2025, I think we really nailed it. Again, keep in mind that the Adelgid populations in BC, I didn't really show that data, but each year we were finding more and more heavy infestations. They don't last long out there. They last about 2 years and then they disappear. But it seems like in the Victoria area, there's quite a synchronized set of outbreaks happening. We think it's because of the warmer climate down there. So a couple of highlights here is we have made two releases that were pretty high, marked by the crosses. Those are redistribution sectaries, much like the first one, but strictly for redistribution. They were done in sites that have relatively isolated hemlock stands. And so we know that the beetles aren't going to get too far there. So we can go and collect some and move them around the province as well. But you can see that we've been not only monkeying around with release levels. A lot of these sites represent different plant hardiness zones within the province. Excuse me. And also, they're on as many river systems as we could possibly find. So you can see that we're really trying to move the beetles to as many watersheds as possible as we do this research program. So in addition to the 9,300, 2,300 also went to Acadia last year for research. So we're starting to build up some really good numbers. We're building capacity, but you know, until we go into these sites and take a look to see if the beetles are persisting, this really is, you know, is a feel-good story to this point. And so what I want to share now is some of the post-release monitoring that we do. And so here's just a summary of a 3-year total. You can see that I think we've got beetles on 7 different drainages now. 7's a nice lucky number for us. And just under 19,000 beetles, 2,800 to Acadia for research. Well, let's take a look at, are these beetles establishing? And so post-release monitoring, I won't read it, but basically every time we put beetles at a site, we go back in for years after to check to see what's happening there. And so that's where we're moving into the post-release side now of this workflow, looking at least I can report now on establishment, population growth, and spread. So for establishment, what we tend to look at is we want to give the beetles a year to establish. So when we release the beetle, we call that an F0. When we go a year later, we're actually looking for F1. So we're looking to see that the beetle offspring survived 1 year in Nova Scotia. And we continue to look at these sites year over year, and that allows us to get an idea of how dense those beetle populations are becoming with time. The other thing that we'll be doing this year, especially, the sites just haven't had enough time to look at, but in this season we'll be able to look at, are the beetles spreading from those release sites? Because that's the other thing that the beetle needs to do for it to be a successful biocontrol agent. And so we do track the weather. This is weather out at Keji. We thought this was our coldest site, but as you can see, using a cutoff of minus 26, we're not getting any days anymore that would really threaten the beetle populations. Now, that's not to say that minus 26 won't kill the beetles, but keep in mind the beetles are up in the canopy where it's a bit warmer in the daytime. We've had temperature probes in the canopy and at ground level And in the canopy, they're usually about 4 degrees warmer. So 22 degrees is really the threshold that will kill the beetles if they're not able to find hidey holes. And I think if you have a warm day that precedes a cold snap like this, I think the beetles can sense this cold coming, and they do get to hidey holes. They usually only venture out when there's a prolonged period of time where they can be safe and feed. So post-release monitoring establishment, we do the same thing because we're trying to recover F1 adults. They come out of the soil, and the first thing they want to do in the fall is hit the first infested branches at ground level. And so, and again, if we don't find it in the first year, we will go back an additional 2 years and look for it. So it's actually a pretty quick story here. The 2023 and 2024 releases all established. Of course, this year we'll be able, in the fall, we'll be able to assess the 2025 releases, but it's looking good so far. Now, again, this is step one, check that box. Step two is, are they still there 2 years after? And so the buildup is where we look at the F2 generation and then continue for several years. But now with some recent research, we've developed a trap that actually collects the larvae. So the larvae are up in the canopy feeding, and they fall to the soil to pupate. And so these traps actually hung underneath the canopy will intercept those larvae as they fall to the ground. So we now have another tool in our toolbox to be able to census these populations. And so what I can say with the, and now there's 6 sites here. I know, you probably remember the 2023 table only had 4 sites. We can include the 2 spring releases done in 2024 in with the 2023 because those were released before that next generation of Laricobius would develop. And so they count as basically as 2023 releases for all intents and purposes. So in this sense, now we can look at F2, except for the Keji insectary. Someone made a mistake and did abundant sampling there. But anyway, as you can see here, the numbers are actually persisting and they're growing. Riverview Park, that one site where we didn't even release 500 beetles, we had one of the highest density of Laricobius per square meter of foliage. Now I want to draw your attention to the bottom of the screen. 25 per square meter is what we get out west at a good site. So we still have a long ways to go. This beetle, in conjunction with the flies, does rapidly reduce populations out in British Columbia. So we're hoping that our numbers will start to get closer to what we find out west. But you can see those are actually pretty good numbers for some of the release sites that we have. Some of the low numbers, I think, are just due to the quality of the site in that first year. And when we started to look around the release sites, we're like, wow, we probably shouldn't have picked this one or that one. Upper Ohio is a good example. The stand to the south is really suffering impacts. And the stand to the north we just happened to find a little paradise that wasn't declining. And so we expect the beetle populations to persist there, but they're probably going to take a little longer because now they're kind of in that situation where the Adelgid population is crashing at times. And so the beetles are going to have to work a little harder to eke out a living in those areas. Mercy River Provincial Park was another one that we had really low numbers, but the release trees were all about 200 meters apart. And that was a bit of a violation of the protocol. We try to have our release trees within 50 meters of each other. And that's the reason for that is so the beetles that we release on each of the trees, so these sites have anywhere from 2 to 8 release trees. And so we want to make sure that the beetles can find each other for mating. But if the trees are too far apart, then we essentially create 4 little tiny populations. And so that explains that one. But the rest, I feel, are talking with colleagues in the States and some of the work I did down there are actually pretty good numbers for just 2 years into doing this. And so I just wanted to spend a bit of time giving you kind of a heads up of what we'll be doing this year. We're going to continue to look at establishment. We're going to look at the abundance sampling, but we're also going to now use these traps to look at spread. The traps, are quite good. We feel they're ready to maybe put at increasing distances away from those 2023 release sites, and to see if we can detect larvae that are up in the canopy, which would be evidence that the adults made it that far there. The other thing that we want to do is with those traps is if we put those in the release trees, I mean, they're only sampling a very small portion of the canopy, but we might be able to show with time when the larvae are dropping to the soil. And so that's what we call phenology. So we can start to piece together how the beetle is developing yearly in Nova Scotia. The last bit here is what we call emergence cages. They sit on the ground. And we're going to go to the insectary where we have very high numbers. So we release 3,000 beetles there. We're going to put those up, and then we're going to collect the beetles as they come out of the soil. That'll allow us to figure out when beetles are emerging. And then that information can then be used to optimize our trips out west to collect. So if we know the beetles are emerging, normally we go out about the third week of October, but if the beetles are emerging the second week, then we'll have to try to plan things a little differently for this. So 2026 releases, just a bit of a heads up. We realized that in all of this work, we're picking sites that have very low density of adelgids, kind of on the leading edge of the infestation where at all possible. But Lucas, and this is Lucas here in the picture, we were down near Yarmouth last fall and we were heartbroken to see all those poor trees. And the other thing that the Yarmouth area affords us is a warmer plant hardiness zone, so where the adelgid's doing a bit better. And we don't have representation in our research project in that plant hardiness zone. So we thought we would try to establish 3 sites near Yarmouth this year, and then 1 site in Halifax to really get a better set of points in the cooler zones, if you remember the map. So we want to try to get 1 more site up there. But I can say that after this fall, we will definitely be transitioning over to research on Leucotraxis. These are the 2 silver flies that attack that 2nd generation of HWA. And I think I'll leave it there, and I hope I didn't chew too much time.
All good, Jeff. I'll pivot to sharing mine now as well so that I can get into the second portion. Oh, wait, here we go. All right. Can folks see that? Awesome. Okay, so so Jeff just gave a great overview of a lot of the work that's been ongoing with Laricobius. Nigrinus is, as Jeff mentioned, it's native to Western North America, found in BC, but also in some of the Western states. Within Nova Scotia, we also have a native species, Laricobius rubidus, that'll come up a little bit later. And then he also mentioned this Japanese species, Laricobius osakensis, which may be in the pipeline a little later down the line too. But as mentioned, there's been a lot of these species have actually been already released in great numbers throughout the United States. You can see sort of a heat map of those releases happening throughout the U.S., and that includes not only these Laricobius species, nigrinus and osakensis, but also these 2 silver flies known as Leucotraxis argenticollis and pinnaperta. And for most of this, I'll just call them Leucotraxis because they kind of serve similar functions. So looking at this secondary predator, and really where a lot of the attention is being focused now. So, as you've heard, a lot of the work on the Laricobius can be done simply through collecting those beetles out west and then planning to release them in due time in the east. There's not necessarily a requirement to have biocontrol to maintain them, but they can be part of a program where we're actually looking at rearing of Leucotraxis. Leucotraxis, as Jeff mentioned, it's collected in a different way. Instead of actually going out and beating the branches to collect the Laricobius, we actually need to go out in the springtime when the larvae, which you can see down here, are active. and then collect foliage that's infested with Western strain HWA. Now, the problem is that in addition to there being an east and west strain of hemlock, there's eastern hemlock, there's western hemlock. The HWA that's found in the west is a completely different strain than we have in the east. So moving foliage that's infested with HWA from the west to Nova Scotia is problematic. And as such, we need to maintain a biocontainment facility and then rear these flies to adulthood to selectively release them into the wild. And this is also important so that we can make sure that the flies are just the flies, that we're not getting parasitoids. And this isn't something like that can be done like the Laricobius in a hotel room. This requires, facilities and spaces to actually maintain. As mentioned, there are these two species. What is interesting is that unlike the nigrinus, the Leucotraxis actually has strains in the northwest, but also in the east. So we actually have strains of Leucotraxis in the east that feed primarily on pine adelgids, either pine woolly adelgid or pine bark adelgid, feeding on these in the east, and they're distinctively preferring these particular adelgids. And of course, they're in an evolutionary context, there has not been HWA in these areas, so this is what they're naturally feeding on. The western strains of these Leucotraxis have been safely released in the U.S. since 2017. And there's fairly recent evidence that they've been established, although they are very difficult to find in the field after being released. So as mentioned, if we want to release them here, they have to be collected, and there is a stage of rearing that I'll get into as well. The benefit of these flies is that, as Jeff alluded to, they actually are specialists on the second generation, but they will feed on both generations of HWA, so they're kind of useful that way. So we have, again, the Laricobius feeding on the cystins, but the Leucotraxis will hit both part of the cystins and then a good portion of the progredens. So it gives you kind of this 1-2 punch where you have the Laricobius hitting that progredens generation, and then to prevent any sort of rebound like you see here with a higher cystins generation or bounce like Jeff had talked about with the cold climate, well, this can also occur with a knockdown of population and more space being permitted through the Laricobius feeding. So the Leucotraxis silverflies form an essential part of building that. They're complementary to the Laricobius, and again, they work best when in concert with one another. To do this, we're basically mimicking work that was developed by Mark Whitmore at the Sarkaria Arthropod Research Laboratory at Cornell. Mark has been an essential part of biocontrol development for these species throughout the Eastern North America. He's been doing this for years and years and years. And particularly for silverflies, they've been very successful at developing a silverfly breeding program. So last year, Mark was kind enough to host my team down there to help us figure out exactly how to set our lab up like theirs. So you can see they have these big plexiglass boxes that are housed. This is, of course, in a secure facility. It's kind of a greenhouse that's been sealed and adapted for rearing the silverflies. Interestingly, the silverflies require natural light. You need to have natural light for them to rear correctly, and it's useful for collecting them from the cages that way. So again, what they've done is they've gone out west, collected foliage. Probably in the next week or so, they'll be going up there for one of their trips. They'll go out multiple times in the spring, mail foliage back under biocontainment, and then basically maintain those flies in these cages until the adults emerge, and then the flies are actually hand selected to be ultimately released into the field. Now at Acadia, through adopting this, we've now developed our own biocontrol facility. I'll show you some pictures of that in a second, but the idea is that this will help support not only the Leucotraxis rearing, but also the Laricobius, and I'll explain what that means in a second. So here's our rearing facility displayed by the fabulous Angie, who's one of our biocontrol techs. We're very happy because, again, we finally have all these cages up and running. Right now we have about 55 of these cages up and running, so it's a pretty decent capacity to have for this. So we have, again, if you're familiar with the Irving Center at Acadia, so one of the greenhouses has now been co-opted for this biocontrol space. And you'll see that we kind of have this room built into, on the bottom there, into a greenhouse. And it's kind of a clean room. And what's interesting, we actually had a sunroom built in this space because it provides a way of actually keeping insects in as opposed to keeping insects out. It's kept under cool conditions, and again, it's ideal for incubating the flies. So the flies are collected through HWA infested foliage out west. They're sealed and shipped to Acadia. We incubate them. The flies are aspirated, and then ultimately they will be released into Nova Scotia forests. At this stage, we're just waiting to have our biocontainment certification from CFIA. I'm anticipating that absolutely any day at this point. And then we'll have the green light to actually start bringing flies into the facility. Once flies emerge from the foliage here, they're actually aspirated and then put in these containers. And then once you have a critical mass and all the approvals in place, you can actually release these flies into the environment. An interesting side effect of rearing these flies is that at the same time as flies are actually being reared out of the foliage, we see these Laricobius larvae that actually drop from the foliage. So while you might get tens of thousands of flies emerging from the top of the foliage, you might also get another 3,000 to 5,000 Laricobius larvae that drop. So using this protocol, you actually get both. We keep the larvae, we put them in soil, and then they're ready for release in the fall. So questions about biocontrol, of course, is whether it's safe. And as Jeff alluded to, you want to make sure that it works, that it's safe, and that it's not doing things that you don't want. And I'll just mention, we do have other Adelgid species in Nova Scotia. Most of them are not what I would call beneficial. But of course, there is concern about whether these might pop over. And I'll just briefly talk about some of the biocontrol research again, being undertaken by master students, and again in co-supervision with NRCan and other folks at Acadia, looking at different aspects of biocontrol. The first is Sammy Stockdale. She's actually looking at evaluation and optimization of thermal tolerance of nigrinus in Nova Scotia. And this is basically looking at things like supercooling points in different range conditions and whether they need time to ramp up and survive cool conditions. And then how their energy stores are impacted. So if they have a really hard winter, are they, still actually fit and able to survive springtime conditions? Second master student is Lydia Jokius. She's actually looking at sort of a risk assessment associated with the Laricobius, where she's looking at other Adelgids that are present, and then using the wild collected Laricobius that are already present and released in Nova Scotia. She's actually collecting their poop. So basically, she's looking at frass metabarcoding. So you actually collect the poop from the beetles. You see what they've been eating over the last few days, and then that's basically subjected to barcoding to identify what they're actually eating out in the wild. They're also, again, working with CFS to develop genotyping by sequencing to figure out if there are actually hybrids of these nigrinus with the local rubidus species as well. So we have basically a roadmap for actually identifying these beetles in the wild. And then Lydia is also looking at distribution. So Jeff had talked about, if you're putting out small populations of beetles, how likely are they to encounter each other? So she's actually using something called a flight mill. This is where you tether a beetle onto a little carousel, and then every time it makes a rotation around that carousel, it clicks off a magnet. So we can actually measure how far and how frequently a beetle flies in a 24-hour period, so we can help understand, how far will they distribute in Nova Scotia and how far apart can you actually put your populations and anticipate them to establish. We have similar work being undertaken for Leucotoraxis with Anita Feng. She's actually looking at the prey range and distribution of different Adelgids that can be fed on by the silver flies, and looking at ways to actually look at how to monitor the silver flies in the wild, either using these yellow sticky traps or branch sampling, or using eDNA, kind of rinsing branches in water and then subjecting that water to DNA analysis. Lots of public outreach, lots of talks, lots of engagement with local partners as well. So it's been really an interesting ride with this. So we're trying to get the word out as much as possible. And of course, working with folks like yourselves is a great way to do that. Again, we're hoping to expand these predator releases relatively quickly, optimize these guys in terms of their thermal tolerance. As Jeff mentioned, understanding the phenology is important. So if we bring in the silver flies, we may need to maintain them in the lab for a period of time, so we sync up their life cycle from a BC climate to a Nova Scotia climate before we release. And then, of course, continue to engage in risk assessments and monitoring these populations overall. So again, lots of people help pay for this. Lots of students spent a lot of time out in the woods dealing with beetles and trucks and all kinds of fun stuff this summer. So again, kudos to them, and thank you all for your time. So with that, I'll stop a little over time. So I hope it wasn't too long, Matt.
No worries at all, gentlemen. That's great. Thank you both very much. Great to see this work rolling. Feels like we have a long way to go, but a lot of really positive early steps with the success of the establishments, and great to see your facility up and running, Kirk. So thank you both very much. We do have a couple of questions. I think one for you, Jeff from Ashley. Which Riverview Park were you referencing for your release sites?
That's top secret. I can't share that.
There can't be that many of them out there, I guess.
No, it's the one that's basically upriver from Bridgewater.
Yeah. Great. Another question from Maggie. Will you avoid releasing the beetles and flies in stands that have been treated with insecticides? If you do release them in sites that have been treated, how long will you wait after the chemical control treatment before releasing the biocontrols? I mean, I think that's a great question because it kind of speaks to the integration of a couple of different tools, and yeah.
I think, yeah, I can. I think the key is to be able to track both the data for the predators and also where the treatments are happening, and then make some timely decisions when we see that the population of beetles is bearing down on treated trees. We know that pesticide will last 5 to 7 years. There seems to be about 5 years of residual activity, and then about 2 years before the population of the adelgid builds back up. So there's about a 5 to 7 year window. Now, as you know, with the treatments, not every single tree is treated in the stand. They're treated on a acre limit for the pesticide. And so there are plenty of, at any given time, there will be plenty of trees in those stands that will probably have a very low dose of pesticides. The predators will forage, I think, in a way that would optimize their opportunities too. At least that's what I think on paper would happen, in that they would, potentially stay to trees that have high populations of live adelgids. There was some work done on feeding by the predators on dosed adelgids, and they don't die. The predators don't die when they eat an adelgid, but they do get a little sick, and they do recover. So hopefully, in those situations, maybe the beetle can learn to avoid, hot trees, I guess.
Yeah, get that question a lot. So that's that's really, really interesting. I see a comment from John advocating a bit for some Digby County sites. Hope you may add one on the Sisebou River.
Well, the Sisebou was one of our field sites a couple of years back. We did a site at Sisebou Falls that received treatments, and that was with John Sweeney, and we were actually discussing this just after the last series of presentations, whether we should go back and revisit it and see where things. I mean, I know how the control plots are doing. There's nothing left, but whether the treated plots might be worth having a look at, sort of the biodiversity and the things there too. It's sort of a 5-year window out now. It'd be kind of interesting to see. But I'm sure there's other spots on the Sisebou as well. So
give me a call.
Thanks. I see another question. We have 70 acres as part of the Working Woodlands Trust. Spring, there'll be treatment. There's no evidence of HWA at this time. At what point could a biological control be applicable post-chemical treatment? Yeah, so you would, Jeff, you would sort of be waiting for the effect of the product to be waning off and the populations of HWA to kind of be building again as the optimal time there.
Yeah. And so we do have ways of monitoring those situations. And, it's a matter, I think, of building a list of potential sites. And I could see maybe moving forward that there would be, if the ground is under like a permanent easement, maybe we can keep in mind the research program on Crown land, but there's no reason why this couldn't be expanded. That's really at the discretion of Kirk's lab. Will be producing these big levels of predators. The other thing that doing what we did accomplishes for Kirk is now we have beetles established in Nova Scotia, and so we can begin to look at what happens when you put flies in some of those populations. So to, down the road, building some research with how the three predators will interact, I think is quite interesting. And I think the pesticide treatments are another thing that we could begin to integrate into all of that. So, we only have kind of on paper suggestions, but I think we could certainly put some hard data behind that moving forward.
One more question here. How far will the insects travel from where they are released? What are you seeing in terms of sort of dispersal or some of those interactions between trees, Jeff?
Well, this isn't any work that we've done yet, but there was work in 2012 done out of Virginia Tech. And within the first 5 years, you're getting an average spread of 80 meters from the sites. So about 400 meters after 5 years. But they do occasionally find beetles much further away. So it's, these are probably the outliers in the overall distance, but there will be some long distance spread by the beetles. And of course, I think the flies are probably very, very good at moving through the landscape. They're very quick. So
I'll add a comment here, and Jeff's done more of this than I had, but sampling in BC, a lot of, you're not sort of in deep forest. A lot of it's in urban areas where, people have hemlocks in their yard, or it's on a boulevard or something like this. So there could be a hemlock that has HWA, and then there's, or one or two hemlocks, and then there's nothing, for potentially miles, right? It just seems like this isolated population, and the beetles are there, right? And they found it. And to me, that strikes me that, they definitely have an acute sense of being able to locate these populations and whatnot. Now, again, the specifics on the dispersal, that I think the jury's still out on some of that. So
sounds like it's just a matter of getting enough of them here so that they are kind of going to become a factor on the landscape. Yeah. We'll take one last call for questions. Appreciate everybody hanging in with us. We covered a lot of ground tonight. I think that was very good. So last call for questions. And thanks everyone. And thanks again to Jeff and Kirk for taking the time. One more question. How do things progress over the next 5 years? Sort of what does the next 5 years look like for us? Hopefully under the best case scenario.
You want to take that, Kirk?
Should I bring up funding in Nova Scotia? We have funding in place for a couple more years to maintain the biocontrol, but I mean, realistically, this is something that needs to be sustained for 10 years to really see impact. And, again, there are ways that we can accelerate this, like Jeff was saying, with redispersals of existing populations that establish, but particularly for the flies, we need to have things up and moving. And again, we are actively pursuing funding to keep that going. So the idea is that as soon as we have everything in place for the flies to go, it basically becomes there'll be research done alongside of it, but it becomes an operational program at that point where we're basically releasing animals every year in a steady flow. So things will accelerate in my mind.
And keep in mind that we need all four of those predators. I think we could probably make a good case with three of them, the nigrinus and the two flies in Nova Scotia at reasonably high levels before we can say whether or not, hey, you know what, this is working or it's not working for the reasons that both Kirk and I lined out with the vagaries of the HWA response to mortality events. So I would say in the next 5 years, I think we're going to see beetles more widely established across Nova Scotia. And hopefully we'll have at some of those sites some burgeoning fly populations, I guess. I'm being very optimistic. So.
Oh, that's great. Yes. Well, yeah, thanks again, everybody, for this. And we will have in 2 weeks, I think, is our final webinar that is going to be touching on silviculture as more of a mitigative, I guess, mostly going to be talking about the mitigative potential, but going to be also touching on some of the research that has gone in here in Nova Scotia, looking at the possibility for thinning is to help increase resilience as well. And hopefully touch a little bit about sort of how to stitch all these together. And certainly don't have any answers there, but can look at some of the work that has happened in the States on how to integrate all three of these approaches over the landscape over time. So with that, we'll say thank you, and hopefully we'll see some folks again on the next one.
All right. Thanks, everybody. Thanks, Matt.
Thanks everyone. Bye-bye.
See ya.